Commonly used for feeding animals, burning and baling, straw is known as one of the 3 Canton treasures. What many don't know is that it can also be used as a raw material to produce biofuel. The increasing demand for biofuel in recent years has prompted the use of cellulose from non-edible plant materials like sugarcane leaves, corn stalks, and rice straw in the production of bioethanol, a clean and renewable biofuel traditionally produced by fermentation of sucrose from sugarcane or glucose from cornstarch. However, since cellulose is cross-linked with lignin in plant cell walls, it is very difficult to release glucose from it. To tackle this difficulty, a collaborative research was started 2 years ago by plant biochemist Dr. Clive Lowe and a student Dr. Lydia Lam from the University of Hong Kong, together with Kyoto University lignin specialist Dr. Yuki Tomimatsu. The research team has recently revealed a new strategy to allow cellulose in rice straw to release fermentable sugar more efficiently. This breakthrough was published in a notable plant science journal, Plant Physiology. Lignin is a complex polymer which functions to provide mechanical strength and structural integrity in plants. However, expensive and complicated procedures are required to loosen the lignin barrier in order to utilize cellulose more efficiently during the production of bioethanol. Rice and other cereals belong to the grass family. Lignin in their stems and leaves contain a special component called trypsin. According to the research team's discovery, when flavin synthase 2, FNS2, a key enzyme involved in trypsin synthesis, is knocked out, not only is trypsin not produced, but the lignin content in rice straw was also reduced by approximately 1/3. In addition, the glucose yield is increased by 37% after cellulose degradation without any lignin treatment. The glucose so released can be used for bioethanol production. In other words, it is more efficient to produce ethanol from this kind of rice straw. The lignin treatment costs can be reduced while the production of biofuel can be enhanced.
This is the first demonstration of the reduction of cell wall lignin content in rice straw by the disruption of trypsin production. Importantly, there are no negative impacts on rice growth and productivity, as plants in the grass family all contain trypsin-bound lignin. This strategy can be applied to other cereals like corn, wheat, and barley, as well as grass species such as sorghum and switchgrass, cultivated around the world exclusively for ethanol production, so that they can be utilized more efficiently as raw materials for biofuel.
I'm very delighted and honored to conduct a research project that could benefit society. Also, as a Hong Konger, I'm always trained to work quickly and efficiently. During the 8 months research experience at Kyoto U, I was particularly impressed by the students there. They perform experiments with extreme care and precision. When I'm doing research today, I always ask myself to do it better than perfect, in addition to seeking speed and efficiency.